The taxon that we are looking at this week is the phylum Echinodermata. Echinoderma literally translates as spiny skin, and that's what these animals typically look like. And we know them as starfish and sea urchins mostly today, but there are also some other odd ones that still live today called crinoids. And there's a bunch of the basic facts of these things here, and a couple of the big ones that are very important. They have a calcite skeleton, and they have pentaradial symmetry. So they have five-fold symmetry. So look at this starfish with its 5 arms. Here's a fossil one with 5 arms as well. So instead of bilateral symmetry, it is what we call a pentaradial symmetry. Besides the starfish and sea urchins, we also have these creatures here which still have the pentaradial symmetry, although in multiples of it. But instead of being free-moving creatures like starfish and sea urchins, they are anchored to the bottom by a stem or a stalk. These things would be suspension feeders, but the starfish are carnivores. Sea urchins are voracious grazing creatures. Very, very interesting group. I have also included in your Canvas module this week a link to a 14-minute video from the wonderful Shape of Life series on the echinoderms, which does a great job of talking about their biological features. I think it's titled The Ultimate Animal, and they really are. Very, very interesting group. I very much recommend you watch that video. Well, let's talk about these different groups from the perspective of a paleontologist and what you may or may not be able to collect or deal with when you're doing your paleontological work. So first of all, let's look at these stalked ones, the high-level epifauna, and there's several different groups. These 2 lower groups, don't worry about those. They're very important in an evolutionary sense, but they're very, very rare. You're not going to run into them. The other 2, crinoids and blastoids. See, here's a stalk. It looks like there's some little rooting structures, then a bulbous top, that would be the head, and then little tentacles or arms waving off of that. So let's do crinoids first. This picture here is from a type of crinoid preservation that is extremely, extremely rare. Look at this. This is beautiful. The stem is here, the whole head is here, all these things. You're typically not going to find something like this, but they're very useful for telling us about the basic morphology of crinoids, which is this. The lower part is called the stem, or the pelma, and then the radix is where it's rooted. The upper part is called the crown, and it consists of the calyx, the head, and then the arms. And sort of tucked within these arms here would be the little mouth. There's a U-shaped gut and an anus right next to it. And these little arms wave through the water column and suspension feed. They grab small food particles and funnel them down to this mouth. There are over 1,000 genera and many thousands of species of these things that have been described. Very, very interesting group. It'd be wonderful to have the calyx and the whole crown area because this is where all of those species are named from. The stem pieces, what happens is this: upon death, the whole animal falls apart. The calyx and the crown usually get completely destroyed, and what gets preserved are chunks of the stem. And sadly, the stem is not taxonomically diagnostic for almost all of the time. And so you can certainly tell crinoids lived here, hell a lot of crinoids, but what species they were, you can't tell. And in fact, you can't tell how many organisms. Every one of these things could have come from the same crinoid, or every one could have come from a different individual. They could have all come from the same species, or they could have all come from different species. Just don't know. Besides crinoids, the other large abundant stalked group or stemmed group are the blastoids. Same general morphology, a calyx with arms on top of a stem, but here the calyx is usually much, much tighter fused. It gets preserved. The arms don't. They get destroyed. The stem doesn't. And so it's sort of the opposite of the crinoid. We get beautiful preservation of the calyx, but not the stem. They usually have good preservation of the upper part where the arms are gone. And you can see the mouth and the anus, the two little holes adjacent to each other. Within this crinoid, then they'd have a U-shaped gut. And so entrance and exit areas are right next to each other. Visually then, beautiful. You can get beautiful specimens of these blastoids. Now let's look at the mobile echinoderms, and they are both infaunal and epifaunal, meaning they have some species that live within the sediment and some on top of the sediment. Starfish and echinoids, and where crinoids and blastoids had a single, simple, conservative feeding style, they were suspension feeders. There's more variety here among this group. So starfish are aggressive carnivores. And if you watch that Shape of Life video, you'll see that. They're very good at what they do. Whereas sea urchins like this, these epifaunal ones, are very good at grazing. And then infaunal echinoids tend to be deposit feeders. These things move by slow motion of their tube feet, which is also shown in beautiful detail on that video. Can you tell that I want you to watch that video? Have a look at it. So first, let's look at regular echinoids. Let's get rid of this. Very spiny creatures like this. So they grow a body, test, a skeleton, and then they are usually covered with a brush of spines. If we look at a cartoon from your book of this, you see how they get the name regular. It's because the mouth and the anus, 19 and 13, are in a direct line, one above the other. So processing of food happens down here. And there might be all kinds of convolutions of the intestines here, but the exit anus is right above it. Okay? The mouth is called the peristome. It has a very unique feature called an Aristotle's lantern. This is a hard part, and you can see this here. But typically, all of this area is very weakly sutured together such that the preservation, if you're lucky, is the outer parts of the shell, but with all of this sort of mouth area busted up and gone. Same thing at the top area. A lot of the sort of area around the anus is also destroyed. Notice as well that all that big brush of spines are gone. Conveniently, though, we are left with a series of little nipples or bumps where the spines were attached. So we can at least imagine the density of the spines, the size, the relative array of them. So let's look at the test or the skeletal morphology, a whole bunch of terms for this. And you can see the sort of ambulacral areas. These are areas where the tube feet project and the food is migrated towards the mouth. Here you see all these little bumps. There would be nipples sort of going off of these. Here's sort of a closer view, and you can see we even have sort of breast type terminology. There's the mamelon. It's surrounded by an areole, and then it acted as a little pivot where a spine was attached. And again, you see we've got a lot of specialized terminology for these things. The anal area, the periproct, is a hole, and then it's surrounded by all these individual little plates. All the little plates are named. They're also numbered for people that do the taxonomy of these things. We don't really have to do this. As far as actual preservation is concerned, the mouth, the anus, That's rare. Actually, whole skeletons are rare. Usually you find broken up pieces or plates and then spines. And so in the lab, I passed around pictures of a lot of actual preservation, but I also passed around some specimens that looked like this as well. So this is here. So I would say that they have a fair to poor preservation in general. This is contrasted with the irregular echinoids, which have this general shape. So they're sometimes called heart urchins. You can see because of the overall heart shape, they still have the pentaradial or 5 radial structure. And when we look at this, each one of these little arms, or these areas sort of going off here, these are the ambulacra. If you enlarge them, you see that they are a field of various little pores that are paired like this. And the size, the density, the arrangement of these pores is another key factor for their taxonomy, their naming. But in a more biological sense, these are the places where tube feet emerge. Spell that right. And they help migrate food downwards toward the mouth. Living ones, are also covered with spines, but notice the spines are much, much smaller and far more dense. Look at this. This thing almost looks like it's covered by some unkempt pandemic hair, really. But the fact is that these spines typically, upon death, they dissolve, they get washed away. They're not typically preserved. But we do have preserved the beautiful, beautiful test. On the underside of it, there is the mouth, the peristome, so it's on the bottom. The peristome is oriented such that all of those ambulacra lead to it, so all roads lead to the peristome. Food is migrated into the mouth here on the underside. If we look at the top but back, there is the periproct, or the anus, and the samples, again, that I passed around, some of them have really good examples of periproct and peristome for you to see. The spines you won't see. They're typically gone, but, hold on, let me clear this up. But, again, this is covered by much, much higher numbers of these little sort of nipple areas, mamelons, areole, again, where the spines were. So we don't have them, but there they are. There's their actual preservation. It's actually quite good. This is actually a sample that was passed around in lab, and they are very, very, very well preserved because of their infaunal life habit. They're already buried. So they have a hard skeleton, and they're already buried. They, right out of the gate, have 2 of the things that are necessary for becoming a fossil. Now this picture right here shows the sort of evolutionary origin of these irregular urchins and why we call them irregulars. It's not because they have a heart shape, it's because the anus and the mouth are no longer aligned in a straight vertical line. So way over here, this time is going by here. So this is early in the Jurassic, and time is going by into the Cretaceous, and then here's the Tertiary. And we have regular urchins here. Notice the mouth and anus are in line. And we start developing or evolving the irregular morphologies, and the mouth and the anus are no longer in the same line. And then you see the slow progressive evolution to ultimately what we recognize today as sand dollars. To look back on this one, This view shows neither the paraproct nor the peristome, but the peristome would be roughly underneath the organism where I'm moving the cursor now, and the paraproct is sort of here in the dark part of this photograph. They're not sort of one on top of the other. And this transition occurs in this middle part of the Mesozoic into the late Cretaceous and then ultimately today. Echinoids themselves can be abundant in a given basin or a given regional area in many, many different paleoecological sub-environments. So you've got regular echinoids on the surface here, you've got various debris-covered ones farther out, you've got sand dollars, you've got shallow burrowing irregulars, and deep burrowing irregulars. They're very, very useful for building up an overall paleoecological picture of the environments that you're studying.